I'd like to thank you all for coming to today's How To Talk by postdocs. And today we've got Dr. Dipanjan Basu, who is from the Reyes Mujica lab, where they study congenital, specific congenital disease in children. And today he's going to be talking about growing cancer cells in a 3D system. So go ahead.
Thank you. Okay, hello everybody. I'm Dipanjan. I have been working with Dr. Reyes for 4 or 5 years at the Children's Hospital, and we work on a specific congenital disease in children, which is called congenital nevi. I'll talk about that in detail later. So today I'm going to share with you my experience in growing cancer cells as free-floating tumor spheroids. So what's in a 3D? This came out in Lab Times a couple of years back. 3D is a real deal, not only on the screen, but also in cell culture tissues. So why is that? I mean, why should we study cancer cells, or any cells for that matter, in 3D? So it turns out that 3D spheroids resemble the in vivo tumor more closely. Why is that? Because so if you look at the tumor in vivo, it has differential access to the circulation, where you have the oxygen, the nutrients. So it happens that the core of the tumor is essentially hypoxic because it does not get access to the oxygen and nutrients as much as the cells that are around it, that are close to the circulation. So there is differentiation in terms of proliferation, cell signaling, metabolism of drugs, drug resistance, and it turns out that the hypoxic core of the tumor often contains tumor stem cells that are having the capability of self-renewing themselves. And it is still debatable whether cancer stem cells exist or not. I'm not going to go into details in that, but what I want to make a point here is that 3D tumor spheroids constructed in vitro resemble this situation much more closely than monolayer cells. You have the same architecture inside the tumor, like hypoxic core. You have differential access to the nutrients and oxygen, and the drug resistance profile that it generates will be essentially reflecting the drug resistance profile of the tumor in vivo. So recently, culturing cells in 3D has become very popular, and many cancers have been cultured in 3D in high throughput format, in several different ways. I'll discuss all those different spheroid models in cancer and how they can be grown, how they differ from one another. And I'll also share my experience in culturing spheroids as well. So this came out in bioengineering journal, where they have shown that in a typical spheroid grown in vitro, you have 3 zones. You have a necrotic zone where you have cells dying from necrosis because of deficiency in oxygen and nutrients. Then you have a proliferating zone, which is the outer zone, where the cells, you get mostly differentiated cells here, and they are also actively proliferating because they have access to oxygen. They're actively metabolizing as well. And then you have, in between, a quiescent zone, where you have cells that are capable of dividing, but whenever they have some extra space to divide, they will divide. So you have a gradient, actually, of nutrients, oxygen, ATP, which increases from inside out, and a reverse gradient of metabolites like CO2 and lactates. So this is pretty much the model that has been established. Now, these cells in a spheroid can be having different cell types in them, which are called organotypic spheroids. and it can be made out of only one kind of cell. When you develop this spheroid from the cell line, then you have only one kind of cell. So I'll go into those different types of spheroids in a moment. These are all the different kinds of spheroids as people have been referring to. You can see that people have named them according to their wish. They have named them colospheres from colon cancer. They have like neurospheres from brain, from mammospheres from breast cancer. But the problem is there is no consensus among the different names, because these are the basic types of spheroids: multicellular tumor spheroids, tumor spheres, tissue-derived tumor spheres, organotypic multicellular spheroids. These are all different kinds of spheroids. They have, their nature is different, they're made differently, but the names are same. You see, you have a spheroid here, you have a spheroid here, you have a spheroid here. So they are all named same way, but actually they're different. So I will a little bit touch upon the differences. So let's look into the details of these different kinds of spheroids. The multicellular ones are typically made out of cancer cell lines. Like you have only one kind of cancer cells that, when you prevent them from attaching to the substratum, they sort of round up. It's more like aggregation than making a colony. So you have a single cell suspension from a permanent cancer cell line, and then you actually create a non-adherent condition. Like this hanging drop method is very popular now. I'll discuss a little bit that one. Then you induce the cells to actually become form an aggregate and form a multicellular spheroid. But this is different from tumor spheres. My research actually includes making tumor spheres, so I'll bring one example from my research to describe this. This one actually has tumor stem cells or tumor initiating cells in the middle of the sphere. So it is believed that tumor spheres are formed when the tumor initiating cell in the middle self-renews and gives rise to differentiated cells. So each colony or each tumor sphere has a tumor initiating cell in the middle, and it gives rise to the tumor sphere. It maintains the tumor sphere. So as opposed to the multicellular spheroid, this is a sphere that is formed spontaneously. So if you seed these cells on a basement membrane matrix like matrigel or something, or agarose, which prevents them from attaching to the substratum, they will eventually make sphere colonies, populate the whole plate. So those are tumor spheres, and you can see that these cells are sensitive to serum, so you cannot grow them in serum. You have to have serum-free medium with a specific set of growth factors for that particular tissue. You have EGF, FGF2 mostly, but it can vary depending on what kind of tissue you're using, what kind of cancer you're culturing. And as I mentioned, they actually proliferate clonally. So one cell gives rise to all the different cells in that sphere. Then you have organotypic multicellular spheroids, which are like you get the tumor lesion. It has all kinds of cells. You mince it up and like make it a single cell suspension, and then you sort of induce them to grow in a spheroid, having all different sorts of cells like fibroblasts or endothelial cells, whatever is there with the tumor cells. So they're sort of resembling the parent organ from where they're coming. So they're called organotypic multicellular spheroids. This is almost the same as tissue-derived tumor spheres. Only difference is here they have the clonal expansion like here. So these are 4 different kinds of spheroids so far people have described. Now I will specifically talk about this, touch upon this a little bit, but these 2 are not my experience and also maybe beyond the scope of this talk. So pictorial depiction of making the spheres. A is the multicellular tumor spheroids. As I mentioned, this is just aggregation. So you have the tumor cells or cancer cells, and then you create a situation where they cannot attach to the surface. So this is actually a very less adherent surface that the cells cannot attach to. And sometimes you also use agitation or like rotating flasks, or like you don't allow the cells to settle and just attach. So in that case, they form the spheroid. This is called multicellular tumor spheroid. As opposed to that tumor sphere, you have a single cell or a few cells that gives rise to the sphere, and they proliferate and the sphere grows. So this is clonal expansion. So all the cells in this sphere comes from this initial cell, and these are the tissue-derived tumor sphere organotypic multicellular spheroids. So you can see here there are other cells, including the immune cells and whatever comes from that parent tissue that you are using as your starting material. All those cell types are maintained here. Definitely, this is short-term. You cannot culture, you cannot passage and maintain these over passages. But this you can, and this you can. But normally, this is used specifically for experimental purposes, like high-throughput screening of drugs and all that. So you don't maintain those cells as spheroids. You culture them as spheroids, do experiments. But when you maintain them as monolayers. But this model specifically preserves the properties of cancer stem cells, like self-renewal and clonal expansion. So this is currently a very popular way to culture tumor-initiating cells, or doing tumor heterogeneity assays, where you can actually basically look at how many of these tumor-initiating cells are there in a particular population of cancer cells. So I will talk about this in a little bit. So going back to the multicellular tumor spheroids, as I mentioned, these can be done in several different ways, large scale or small scale, whichever way. The most popular method is this hanging drop method. You can also do a roller tube, where you actually keep the tube rolling with media and cells, so that the cells will never attach to the tube, but they'll come in close contact with each other, they'll interact with each other to make spheroids while they're rolling. And then you can get those spheroids sit into different plates for your own experiments. You can have a spinner flask, you can have gyratory shaker, where you keep shaking the flask. So the main aim is not to let the cells attach to the substratum. Then you have soft agar liquid overlay. This method evolved through this. So people were trying to develop a technique where you just sieve the cells in droplets, and the droplets themselves are hanging, so the cells will never have a chance to attach to any substratum. And they use soft agar liquid overlay. Then there were some plates in the beginning that came out, like you have to sieve the cells and then invert them so that the drop is hanging. But now there are plates, they are engineered in a way that you just have to add the cell suspension in those particular spots, and that creates the hanging drop. And over time, these cells get compacted and they form spheroids. One good use of this is done in the drug discovery research, that you can specifically seed one spheroid per well in these plates in a like high throughput format, like 384 well format or 96 well format, and then you can have several different concentrations of drugs, treatments on that plate, where every well has one spheroid. So the analysis becomes much easier, because you can look at one spheroid, how it is responding to the drug, and you can actually add the treatment directly in those drops. You can add 1 microliter of drug to each drop, and it doesn't change the situation in the plate. So this is very popular now in high throughput formats. And but as I say, you cannot maintain a cell, passage them in this format. You have to maintain the cell in a monolayer format, and then when you prepare them for experiments, you can use these kind of plates, make the spheroid, and do the treatments and stuff. So, tumor spheres. Okay, these are my cells. Now, as I said, tumor spheres are formed from single cell, giving rise to a sphere by clonal expansion. So, all the cells in here are coming from a single cell or 2-3 cells inside the sphere. These are the cells derived from a particular melanocytic lesion of a congenital disease. I will show you that lesion in a moment. As you see, they are brown colored, they have melanin pigment in them, and this is how they look like when they grow in monolayer. They are dendritic, they have a very small nucleus, and when you plate them in a basement membrane matrix or agarose or something that will prevent them from attaching to the bottom of the plate, they will make eventually spheres. These kind of sphere growth was first described from the brain in 2003, not so long back. They named them neurospheres. And from then on, right now, this is the only reliable measure to determine self-renewal in cancer stem cells. So there is a debate about some people on the other side of the spectrum. They say, where is the cancer stem cell? Is there any cell like cancer stem cell? There are markers. There are conflicting opinion about the markers, but the only reliable assay that everybody agrees to is a self-renewal, and that can be assayed in this format, where you seek the cells and they give rise to other cells clonally. So they have the ability to self-renew and give rise to differentiated cells as well in the tumor. So this is also known as tumor heterogeneity assay for a particular tumor lesion or tumor biopsy. So we work on this kind of disease. This is called large congenital melanocytic nevi, and also another part of it is neurocutaneous melanocytosis. Now, this is a complicated name, I know. This little baby was born with this much huge burden of a tumor. This tumor is benign, actually, not malignant. And they're called large congenital melanocytic nevi. Melanocytic because this is pigment cell tumor. And this is called a nevi. You can have acquired nevi from sunburn, which can eventually transform to melanoma. This is congenital. So kids are born with this kind of lesion. This is a very advanced stage of this lesion, but sometimes you have birthmarks, sometimes you have moles, but it's the same nature, but it is a huge burden of tumor. And this particular patient also had a brain lesion that he had proliferations of melanocytes inside his brain and deposits of melanin inside his brain. When the picture was taken, the patient was 1 year, 9 months, and all these cells harbor a somatic mutation in NRAS exon 3, which is Q61K mutation. And these cells, since they're benign, they're not like malignant cells. Mostly the treatment that's available to these kind of patients is surgery. You just take out the lesion and you'll be fine. This has been going on for years now. This disease was first described in 1861. And from then on till now, the only treatment available is surgery. But the problem is, surgery could not provide any cure, because that little kid will eventually get back his tumor. So the question is, what causes the tumor to come back if it's not malignant? So one answer could be our colleagues in France, they came up with this paper recently, that clonogenic cell subpopulations maintain congenital melanocytic nevi, where they actually cultured some cells in a spheroid format, but they don't look like very compact spheroid because they were cultured in agarose. So I will show my pictures, which are compact colonies. But they sort of use this spheroid culture to count and figure out how many of those cells exist in a particular number of lesions. Particular number of tumor cells. So when they took out the tumor cells and cultured in spheroids, like 1 in 250 cells were tumor-initiating cells. So 1 in 250 cells were capable of repopulating the tumor, and they could self-renew. They could express the stemness markers like OCT4 and nestin. They also express drug resistance genes like ABCB5 that pumps out drugs. So whenever you treat those cells with drugs, they are resistant because they have the ability to pump the drug out. So this paper actually prompted us to think that, okay, so if clonogenic cell subpopulations exist in nevi, and the only way to catch those cells is to grow them in spheroids, so why don't we grow them in spheroids? So what I did is I cultured some cells from a lesion of this patient. We actually have IRB approved protocol of receiving freshly excised tumor tissues from these patients. This patient had undergone surgery in Chicago, not sure health system, but they shipped us the sample. And we have a tissue repository in the lab that maintains tissues from all these patients. And we sort of try to culture cells from these lesions and try to grow them as spheroids. And here you see a bright field picture of cells trying to form a spheroid. These are even not compact either, but they are sort of trying to form a spheroid. And they express the melanocytic markers here. So sort of validated that these cells are really coming from the lesion, not from any other. And the way we culture them also eliminates the possibility of growing other cells in the tumor. That I'll come in a minute. I tried many different other patient samples, and they all were capable of growing as tumor spheres. We named them Vivo Spheres now. So this patient, PS01, is the cells came from the spinal cord lesion. This patient, you already saw, it came from the skin. This patient also from the skin. This patient, the tumor cells came from the brain. But they all behave similarly. And when we put them on Geltrex is a basement membrane matrix that has laminin and collagen in it. They grow better than in monolayer. So here I show you that the proliferation ability of those cells when grown as spheroids is way more than when they're grown as monolayers. Okay, so this spheroid formation is not actually a mere aggregation. There is something biological going on inside. That is, this is a proof that E-cadherin, which normally is maintains the contact, cell-to-cell contacts on it's located on the membrane. When you grow these cells as monolayer, they are in nucleus. They don't express. They express E-cadherin. They are not localized on the nucleus. But when you transfer them to the Geltrex matrix, E-cadherin is redistributed to the membrane and cytosol, and these spheroids actually are maintained by cell-to-cell contacts, and E-cadherin is on the membrane here. We can actually count the number of spheroids. So we can compare the number of spheroids present in each patient's sample, and it has perfect correlation with the aggressive ability, the proliferative ability of the cells. And these cells grow faster, more aggressively in culture, so definitely they generate more colonies than the other three. Also, we can measure the diameter of the spheroids using softwares in microscopes. You can put the spheroid colony under microscope. You can measure how the micrometer of the spheroid, the diameter of the spheroids, how much micrometer. And the specific good thing about the whole is you can actually passage them and maintain the same ability over, like I followed them like 5 passages. The number of spheroids remain almost the same. So the self-renewal ability is preserved, and you can basically maintain these cells as spheroid colonies, and you can preserve their properties. And definitely the one which has more colonies over passages, it showed some difference going down a little bit, but all others I couldn't see any significant difference. Okay, here's the good thing about it. So if we can maintain sphere colonies as if we can maintain over passages, we can actually use them for drug treatment or drug sensitivity assays. So we can treat them with drugs and measure the number of spheroids or diameter of the spheroids. Specifically, if the spheroid is not growing, if the self-renewal ability is impacted by the drug, the spheroid size will not grow. So compared to vehicle here, which is untreated, we treated with 4 different drugs. This is vemurafenib is actually FDA approved for melanoma with BRAF mutations. MEK162 is in the last stages of clinical trial. It's not approved yet, but it's showing very good response to NRAS mutated melanoma. GDC0941 is a PI3 kinase inhibitor, and GSK2126458 is an AKT inhibitor. So NRAS mutation has redundant signaling going on, and people have tried to target those mediators. Like MEK is one of them. Another parallel mediator is AKT. So those works are going on. Still, NRAS mutated melanoma has no surefire cure like BRAF. We picked up these 4 drugs, and we sort of tried to see, since we detected NRAS mutation in this disease, whether these drugs can affect the self-renewal ability of these cells. And as you can see, vemurafenib did not affect anything. MEK162, little bit. GDC, same as MEK, but this one turned out to be the most effective, which targets AKT. And then we measured the diameter, and this is definitely very significantly down here. I did not put the significance analysis here, but visually you can tell the difference. This is also effective, but I think this drug is not inducing cell death as this one. You can see a lot of dead cells around. It probably stops the cell proliferation, but the cells are still surviving. So these kind of studies will give us like more reliable data about a drug action, because it's working on the spheroids like in the in vivo environment. So we are working more on this. This is just preliminary data, and we hope to find out more. These are some cell viability assays you can do using the spheroids. The same drugs used here, different patient samples, different spheroid cultures, and GSK turned out to be the best one in every case. Although there are some patient-specific differences. So this is another aspect I'm so interested to pursue, that you can use this as a preclinical model to test drugs before you give it to the patient. So incidentally, these 4 drugs are not, except remurafenib, none of them are approved yet. But if the doctors want to prescribe a drug which is approved, you can basically use this model to test the drug before you give it to the patient, how good the patient will respond to the drug. Yes?
Have they been used in animal models?
They have been used in animal models, because the problem is animal cells, like mice cells and human cells, are genetically not similar. So this is more reliable if you get the cells from directly the patient. You can grow these cells from a very little tumor biopsy. You can pull out using a needle, culture the cells in the lab, test the drugs, and then you can tell the patient, well, this is the drug that works for you.
What's the time frame for that?
2 weeks at least. Because we need to first make the cell survive, then expand, and then go into the spheroid, then treat. So at least 2 weeks, because I would guess that the more aggressive the cancer is, the time frame will become short and short. Since these are not aggressively dividing cells, so the time frame is longer here. Okay, so this is like the summary of what I was trying to say all this time. So how do we grow these cells as spheroids? So we get a tumor sample from biopsy or something. Very rarely we get a sample when a patient is dead. That's very sad, but normally we get biopsy instead of autopsy, and we trypsinize and plate the cells. Like this is a picture after 72 hours, which shows you that the cells are still like shaky. Some cells attach to the bottom of the flask, but they have not yet started to give out the dendritic expansions. So it takes a while for these cells to really go into division. That's what I was mentioning. Like 6 days, you can see clones of cells coming out on the plate, like several areas on the plate where you see bursts of proliferative activity, and those cells are like this. They have short dendritic expansions and not so brown at this point, but the more they grow, like 10 days, they sort of start dividing now, and they become confluent in 21 days. And then if we have enough cells, then we can trypsinize them, put them in the matrix where they will round up. This is lower magnification. It's not the same as this. This one is. I just wanted to show you how many we get from like a clone of cell. They round up like sphere colonies, and then this is how they look like. One colony looks like this. You have like cells kind of radiating out from the colony. Those are the differentiated melanocytes, and I would guess the cells in the central part would be more or less differentiated, kind of. So that's the timeline, and once we get this, then we can now seed them for experiments. We can study drug sensitivity or cell signaling or whatever we want to do with them. Immunofluorescence. One problem with, not a problem, but I mean the ways we harvest cells from monolayer cultures and the way we harvest cells from spheroids is different. So for cell survival assays like MTT and all that, you can directly do in the plate. You can add the reagent, and it's like colorimetric, so you can wait for the change in color. But if you want to do Western blot or something, if you want to harvest the cells, then normally you have to get rid of the matrix first. So you solubilize the matrix in PBS, EDTA or something like that, and then you pellet down the cells, then you lyse the cells and go for. So it's not very. I mean, you cannot really get an exact count of cells in every sample. You have equal number, but what we do normally is we estimate the protein content and then normalize with that. It works very well. So here's some useful resources that I used and helped me start this method. Definitely, this is a very good book, 3D Cell Culture by John Haycock. And the Geltrex matrix, if you go to Life Technologies, they have different kinds of matrices that are used for different purpose. Like they have embryonic stem cell qualified matrix, they have virus-free, growth factor-free, different kinds of matrices. So we sort of, our choice of matrix was based on previous efforts of other groups that tried to grow melanocytes. They found that having laminin and collagen increases the survival, so we used that matrix that had the laminin and collagen in it. Also, 3D Biometrics is another company that recently they have a lot of protocols on their website that they're free. You can just go through and use them. They also sell the plates I mentioned, multicellular tumor spheroids, hanging drop method plates. They have those plates as well. I don't use them, but I mean I found there. So those are some useful resources that you can use if you want to grow your cells in spheroids. And this is our group. This is my boss, and these three students are from Pitt, first experience in research. They started this year, so they worked with me, and they did some projects. So that's all for today. Thank you for your attention. I will be open for any questions. Yep.
So you did the drug assay with the 3D microspheres. So I was wondering, the spheroids, so I was wondering, did you actually do a comparative assay also with the monolayer to see how the variability of the drugs?
Yeah, that's a very good question. Yeah, we actually did. And there is, so the way we are growing these spheroids, we are actually selecting the tumor initiating cells in the very beginning when we are growing in monolayer, because if you go back to this slide where I summarizing the methods, so what happens is this, between this time, 72 hours, here you have all kinds of cells, but at 6 days, all the other cells like fibroblasts and blood cells and whatever comes from the tumor lesion, they're all dead. The media we are using, having these specific set of growth factors, only supports the growth of these cells. So basically, if you take these cells now and do the drug treatment assay, the data that you receive, and if you take these cells now and do the drug sensitivity assay, there is not much difference in terms of viability. But there will be difference when we look at interaction with the matrix or cell signaling or other parameters. So, yeah, that's a good question that reviewers asked us. Why don't you use the monolayer then? What is the use of using spheroids? Yeah, I mean, there is no difference in terms of viability. But definitely, if you delve deeper into other intricate questions, then there will be differences. There will be advantages of the spheroid model.
And I was wondering, because it's in the spheroid, so you could probably keep it longer as opposed to a monolayer plate where, because the conference is so you have to
you're right.
finish the drug assay within 3 days.
Yeah, if you keep changing the media, you can really keep them longer.
How long?
Yeah, I have kept them like 2 weeks, or I mean more than 2 weeks. So what happens is, if you look at, again I'm going back to the pictures. If you look at these cells, so the number of spheroids will not increase, but what happens is, these look at how these cells are touching each other.
Yeah.
So as the spheres grow big, they seek out their partners. They're very interesting cells. They want to touch each other. So as they grow, they come in the vicinity of the other neighboring colony. They sort of touch each other. So I stopped there. I mean, you can still go on. I mean, it's no big deal. But the problem, another problem is the core cells will start dying from. Like I mentioned, the hypoxic core. Yeah. As we keep going, the colony will grow big to a certain extent, but then it will start dying because the core cells will die.
So when you're doing the viability assay, were you looking at viable spheres and viable cells?
I'm looking at viable cells. It's synonymous. I mean, because spheres are made out of cells. But what I do is I do two things. One is I directly do MTT assaying in the well, add the MTT reagent and wait for the color development, measure in a plate reader. And the other things, parallel plate, I give it to my students that you trypsinide and count the cells. So you see if it matches. So it sort of matches. Definitely more human error comes in counting cells, but sort of it's in the same ballpark. Yeah. Yes?
Is there any difference between the gene expression between when you compare in a spheroid form versus monolayer form?
Oh, yeah. So I cannot give you the data from my cells because we haven't done that in these cells. But in other cancers where people have grown monolayer versus spheroids, like Hans Clevers in Netherlands, he was the person who made this organotypic colospheres from colon cancer. They did like a whole transcriptome analysis of monolayer versus the spheroids, and there are many genes that are up or down regulated that sort of gives them an idea how these cells. So they did like the parent tumor, monolayer, and spheroids, and then they compared all those three, and definitely the spheroids are closely resembling the parent tumor. But we haven't done that yet. We are still starting on that. Yes?
So as I understand, your real goal is to characterize the TICs, but what if your goal was to really kind of recapitulate the behavior of the tumor in vivo? Would there be benefit to not selecting out all those other cell types, but maybe formulating the media or whatever your modeling matrix, so you could really regrow like the 3D structure with the different cell populations that you might have originally had? Like, was that done in the rubric?
Very interesting question. Yeah. So in the beginning, when we were toying with this idea of growing these cells in spheroids, basically the problem with this, the difference of these kind of cells with other cancer cells is that, so the key thing here is the tumor heterogeneity, as you mentioned, that you need to have all the cells. Each one has its own contribution in the tumor. But the problem with these cells is that the huge lesion you saw, most of the cells are senescent. So they don't divide. Only if a few of them divide. That's why if you have a child with a huge lesion of melanocytic nevi, it according to NIH, it poses little threat to the child because the child will not die from it. But what happens is at puberty, the tumor starts growing. If you do surgery, the tumor comes back. As you grow older, you run the risk of having melanogenesis from those tumors. So there are a few cells in there which are capable of dividing. And those cells are at risk of acquiring secondary mutations and transforming into melanoma. So our target were those cells. But other cells, definitely having those senescent cells could have some implications in the tumor microenvironment, but we cannot target them because they don't grow in culture. They are senescent. They're like permanently growth arrested because of oncogene activation. Only the cells which have self-renewal ability, they keep dividing, but they are also not very actively growing cells. If you provide them the environment, then they grow. They're like quiescent, but they start dividing once they have all the ingredients necessary to divide. So it's kind of an interesting system.
So you're showing a picture of this patient. That this patient actually also had some lesion in the brain.
Yeah.
Due to the metastases, or despite of that, actually the patient is surviving?
I'm not very good at reading MRIs, but I just put the picture to show they have a lesion in the brain. So the precursor cell of melanocytes are neural crest cells. The neural crest cells are cells when the embryo forms and invaginates. Before the neural tube forms, you have the neural crest. Those cells are the stem cells which give rise to many different cells in the body, including melanocytes. Now it is believed, not proven yet, that those cells, when they start migrating from neural crest and during their journey to the skin, at some point during development they acquire the mutation. And when they acquire the mutation, they start proliferating like abnormally, and then at some point they stop because of tumor suppressor mechanisms in the cell. So you have this benign lesion. Now, the later in their journey they get the mutation, the phenotype is mostly skin. The earlier they get the mutation, the phenotype is like brain plus skin, something like that, but it's not proven yet. There are some lineage tracing studies in chick embryo. Nicole Duran, the stalwart of neural crest biology, her group is still working on this. But I mean, those are genetically modified, GFP tagged, and there people argued on that model that it does not recapitulate exactly what the migration path is. But definitely, the patient that we encounter are born with both brain or skin. It's not like they metastasize to the brain. But definitely, some patients with both lesions have shown metastasis where the cells end up in lungs or kidney or some other vital organs. But when they have the neurocutaneous, that's what the name is, they have both. They're born with both the components of the thing.
How are they cognitively?
There are some developmental delays reported for these patients. So we have one patient who is 16 years old, still living, with no apparent skin lesion left after surgery. The brain lesion, since was benign, the physicians recommended that we don't go for surgery yet. We'll keep a close eye on it. She had no apparent symptoms, but developmental delay. But they show like vomiting, a lot of different symptoms related to brain, like seizures and a lot of different, depending on the severity of the tumor size, what areas it is pressing and all that. Hydrocephalus is another symptom where they have water inside the brain. Different kinds of symptoms, but mostly they show they all show developmental delay.
But it doesn't target anywhere specifically in the brain. It can
No,
just be all over
no,
the place.
no. Leptomeninges is the most common known area where probably during development that's where the neural crest was. I mean, I don't know. I'm just curious. But leptomeninges is the most like here, and also you see these white things are actually the melanocytes. Yes, ma'am.
So there's actually a mutation in the DNA? Do you know what causes the mutation?
Yeah, it's a somatic mutation. It's not a germline. It's just those cells have the mutation, and they clonally expand their population, so all those other cells have mutations, other cells coming from those cells. But you don't have it in the germline. So if you take blood DNA and look for that mutation, they'll be negative.
So NRAS is the only mutation you found, or any other?
No, I mean there is BRAF also. So we showed BRAF, and NRAS is from our colleagues in UK. I mean there is like a consensus now that NRAS is the most predominant one, but there is BRAF also. It's also GNAQ. There are a few mutations found, but they are the rarer ones. The predominant one is the NRAS. You have Q61K, you have Q61R, two different kinds of mutations of NRAS, most common in these patients.
Any other questions? All right, well, thank you very much.
Thank you, my pleasure.